You're listening to Let's Talk AI.
Welcome to Let's Talk AI. I'm Harold Godwin, Managing Director for Waterloo AI, and I will be your host today. Our guest is Scott Hopkins, Associate Professor in the Department of Chemistry here at University of Waterloo. Welcome, Scott.
Well, thank you very much, Harold. It's a pleasure to be here.
Awesome. So you've got so many exciting research projects and things going on, but before we go there, let's go back to where it all started for you. If you could share with us your background, where you did your undergrad, master's, PhD, and then how did AI kind of get embedded into all that?
Get all mixed in there. So I started my undergrad back in 1997, previous century, at the University of New Brunswick, studying chemistry. And from there, fast-tracked into a PhD program. So I skipped the master's. And my PhD was in molecular spectroscopy. So I was basically using light to look at the shapes of molecules, figure out how the atoms were bound together. After doing my PhD, I then undertook postdoctoral studies at Queen's University in Kingston, Ontario. In this case, I wasn't looking at the structures of molecules, but how I could blow them up with lasers and see what pieces went flying everywhere. About 18 months after starting there, I moved to the UK, where I did postdoctoral research at the universities of Cambridge and Oxford. And in that case, I sort of mixed those two ideas together, my PhD and my first postdoc. And I started holding things in electrostatic traps in vacuum. So sort of like what you'd see out in space. And then blasting the heck out of those molecules with lasers to get the bits to fly off. And the tricks here are to find out what bits fly off and in what directions. And the whole goal of doing this sort of work is to kind of get a quantum fingerprint of matter. And by doing that, you know without question what the properties and structures of molecules are. So in amongst all of that, I started learning some chemical theory and doing some computer programming. So when I moved to the University of Waterloo in 2011, I started my own lab working on these sorts of ideas. And after a few years, around 2016, 2017 or so, it became very clear to me that the processes that I was dealing with were so complicated and that I was generating so much data that I could no longer use my organic neural network, my brain, to actually figure out what was going on anymore. So I had to start introducing artificial intelligence, machine learning, to try to make some sense of these reams of data that we were collecting. And following some hiccups along the way, since it did take a little bit of time to climb that learning curve and actually become functional back in those days. This was before you could download a nice package in Python to run things for you. We started developing some really nice predictive models where we could take the structures and the behaviors of our molecules in our experiments and start correlating that with physicochemical properties. So these are things like how easy it is to dissolve a molecule in water, how quickly it can move through the human body, things along those lines. And that tied us in with doing, say, preclinical drug development in collaboration with some pharmaceutical companies. We started looking at that in the context of environmental contamination, right? How contaminants can move through the environment, how toxic they might be to different organisms, people, fish, things along those lines. And from there, it just sort of blossomed, right? We're now developing predictive models for all sorts of different things and introducing things like SHAP analysis, which is sort of a newer method of interpreting complex models so that we can actually understand the processes that we're measuring experimentally.
So that brings us up to kind of where you're at today, I'm guessing.
Exactly. Yeah.
So what is your lab called? How many people in the team there? And what's the big goal you're chasing now? Can that be summarized? Is there a big goal, or is it general still?
We've got a couple big goals that we're chasing. My lab is very creatively called the Hopkins Laboratory at the University of Waterloo. So, we can't claim to be very creative in our endeavors with naming things, but certainly in the scientific sphere, we are. Um, Depending on the year, I mean, the people in the lab depend on funding and scholarships, and you get that natural cycle with some people graduating and then having to replace. But we're normally around 10 to 12 people that work in my group, ranging from master's, PhD to postdoctoral fellows. And again, depending on the year, we actually have undergraduate students come and work with us as well, doing co-op terms or honors research projects in the last year of their undergraduate degrees. And I guess the main thrust of what we're doing right now is on the technical side. What we're doing is actually measuring how molecules behave in dynamic electric fields. So what we're doing is subjecting a molecule to a really high frequency, high voltage electric field so that we can see how it behaves, which sounds very esoteric, but what it turns out to be useful for is we're actually tuning how that molecule can interact with its environment by doing that. So when our molecule is under low field conditions, it's actually moving very slowly. And when molecules move slowly, they're cold. When the fields are turned up to very high field conditions, the molecules get accelerated. They're moving very fast. And what that means is we're effectively heating the molecules up and doing that with electric fields, which we can really control well. So by monitoring how that happens, we can actually use machine learning to correlate that behavior with a whole raft of interesting properties. And we've been recently looking at things like how molecules can pass through cell membranes, but not needing cell membranes at all to do this. All we're doing is jiggling these things around in a high frequency electric field, and then using machine learning to tell us exactly how that would behave in, say, your kidney cells, right? Things along those lines. So the broad goal of what we're doing is just trying to understand how structure and molecular properties relate to one another, but in a sort of a specific fashion. We've been really focusing recently on things like metabolites. The things that your body generates as it works, and in screening metabolites, you can actually deduce and diagnose metabolic diseases. So, for example, if your liver isn't functioning well, your body is going to produce different molecules than it would if your liver was functioning well. And we can separate those different systems and use machine learning now to develop a model to say, oh, you've got a liver dysfunction, for example, or you might have early onset glaucoma. You'd better go check with a medical specialist on these things.
So it's the metabolites that are the indicators, if you want to call them.
Exactly. So we would call those biomarkers, and they're basically little flags that your body is throwing up saying, oh, something has gone wrong here, something is malfunctioning, and now we're generating this thing instead of the stuff that we would normally want to make.
And is this entirely new, or have these biomarkers been noticed or identified previously?
So some biomarkers are well known, and others are not. In the metabolic area, for example, humans have something on the order of 200,000 different metabolites, which means, I think it doesn't have to be stated, humans are very complex systems, right? And if you want to have a molecular map of a human being, there's a lot of things that you need to consider. Some of those are endogenous, things that we have inside of us. Other things are contaminants that come from our environment. And this is another area that we're actually working in right now. You probably have heard of something called a forever chemical or forever chemicals. These are fluorinated species known as PFAS, per and polyfluorinated alkyl substances. There was a really good movie on this a few years ago, actually, called Dark Waters, and it's basically outlining the use of these fluorinated chemicals to make all sorts of really useful products like firefighting foams and food packaging and nonstick cookware, things along those lines. But the molecules themselves are actually being linked to a whole range of diseases in humans, running from cancers through to metabolic disorders and thyroid disorders and things along these lines. So on the bright side, here in Canada, we don't make any of those molecules. Those aren't produced in Canada, but we import them in many different goods, right? So if you've got a waterproof rain jacket, there's a possibility that these molecules are embedded in that waterproof rain jacket, and they are appearing in the environment, moving through the environment. They are very chemically stable and resistant to degradation by physical or chemical means, and that's actually a problem because when organisms eat them, right, through either plants that are contaminated or other animals that have eaten plants that are contaminated, it bioaccumulates, meaning the concentrations build up in your body faster than you can excrete them. And now you're approaching concentrations that can actually cause health problems.
Is that one of the impetus behind, the air conditioners and the restrictions on the fluids they were using in air conditioners?
It's not the same molecule, so I think you're probably thinking of fluorocarbons,
Like the freons
CFCs.
and some of those things.
Exactly. So these halogenated molecules have been linked to problems in the past for other things. CFCs, if I remember correctly, are carcinogenic, meaning they have been linked with causing cancer, but they're also a real problem for the greenhouse effect and global warming, right? And that, I think, is now becoming a very evident problem around the world. You can certainly see it in Canada these days with the forest fires and the heat waves that we're having, and on the East Coast, et cetera, et cetera. So this all sort of underscores the reason why we really want to know the structures and properties of molecules before we go into mass production and distribution of these guys, right? Because it can be a complicated process when you're emitting those into the environment.
So you talked a bit earlier about using machine learning to do this. Take us down that rabbit hole, kind of a little bit more insight into exactly how you're using AI.
So originally, it was pretty simple supervised learning tasks that we were doing. We would start by saying, okay, we've got this molecule. Here's some of its properties, its mass. Here's how it behaves in, say, a mass spectrometer, which is one of the instruments that we use to measure the behavior of these molecules. And we would say, given that information, and knowing that it has some degree of solubility in water, can we now make a prediction on another molecule that's similar? And of course, the I guess rule of thumb for machine learning with supervised tasks, anyway, is that you need to have a fairly big representative data set so that you can build some sort of predictive model. So for many years, we were essentially turning a crank and generating as much data as we could. In that process, we're trying to study interesting chemical species so that we can learn something about their quantum structure. But always in the back of our head, we're thinking, hey, we're making a really nice library here. How are we going to address this with a global perspective? And that's really where the machine learning aspects came in. So in addition to that sort of supervised machine learning methodology, which has evolved over the years, right? We started with, simple decision trees and random forest methods, things like this. We've now moved up to some more modern methods, extreme gradient boosting, stuff along those lines. But even that is not cutting the mustard for a lot of things now. And more recently, we've started exploring transformer architectures, like as are used in ChatGPT. In this sort of scheme, your transformer, in a nutshell, is not only drawing relations between, say, adjacent words in a sentence, but in terms of the context of words in the wider sentence, so that you can have almost like a language understanding and processing ability, right? So anybody that's used something like ChatGPT will know that you can ask it a question and it will give you an answer that sounds like there's somebody on the other end of the line typing the answer out to you. In the chemistry context, we can actually map that process one to one. Because if you think of a molecule, each atom in a molecule is almost like a word, and the arrangement of those atoms in the word, and not necessarily just the ones next to each other, actually inform the properties and behaviors of that molecule, right? So if we have whatever, 2 carbon atoms side by side, then bound to an oxygen, that's going to be very different from a carbon bound to an oxygen bound to another carbon, right? So what we're doing is employing a language model to interpret the language of chemistry. And by doing that, we can then make some predictions of observables, right? So for example, what happens if I shine light on this molecule? What happens if I give it a really big smack with another really high energy particle? Things along those lines, and make some actually very accurate predictions of that behavior, which we can then go and test in the lab to make sure that we're on track. 9 times out of 10, it actually works. And that 1 time out of 10 where it doesn't, we can then raise our finger and go, aha, what are we missing? Right? And then that helps direct our research down those paths.
Okay, so this sounds like really core foundational kind of research. What does the future look like? Where do you see this going if you could connect the dots forward to paint a picture of the future?
I think what we're starting to see right now is that the way that we're leveraging machine learning is actually highlighting gaps in our knowledge. So my opinion is that you're not going to be able to replace the researchers, the people themselves, because we're able to synthesize things that you might not normally connect, right? So I think we've all had that scenario where you're just falling asleep or just waking up, and 2 thoughts that don't seem to connect all of a sudden do. And it's like, wow, I never thought of the world that way before. But I think AI as a tool for identifying gaps in knowledge and directing research into really interesting areas could be a really neat way forward. And it could start tying together things that you don't normally expect, right? Maybe, biology and quantum information processing, throwing out totally random examples here. But that's one direction I could see going. Another direction that I could see being very useful would be in meta-analysis, right? There's a lot of information out there right now held in lots of different databases, some public, some not. And one of the issues that we have underscoring advancement in science, or rather inhibiting advancement in science, is the issue of reproducibility. So for those that haven't heard of, quite often, certainly at frontier science, we seem to run into an issue of being able to reproduce results from one lab to another, or from one instrument to another. You'll see a study published where people generate some really good, interesting results, and then somebody on the other side of the world tries to reproduce it, and they're unable to, and we don't know why, right? Quite often it comes down to very simple things that we just don't consider. For example, maybe the humidity in Waterloo is too high so that we can't reproduce results from someone who's done some measurements in Arizona where it's really dry. and that's just not something that was on our radar to look at. But if you can take a global view of all of that information, which is not something you can do with your, again, organic neural network, right? That's something that you need big computing power for. Then you can start teasing things out to say, okay, so if we look at this from a different perspective, you can see actually these are two different experiments, even though they look the same. So if you want to actually test reproducibility, here's the thing that you need to do.
Okay.
Right.
So said the term meta-analysis. In real simple language, what does that mean?
A typical scientific analysis that I would conduct would be me taking a bunch of measurements to get some data in my lab, and then working on that small data set, I would then try to draw some conclusions and make some predictions for future work that could be done. Here are some open questions. Everybody does that, right? But what very few people do is to collect data from labs that may be working sort of in a similar area from around the world and look at all of that collected data together to see what's consistent and what is not, right? The reason why we don't do that is, one, there hasn't been a big push to do that, although I think that's coming. And the other reason is science is pretty expensive. So, my lab to create, tens of millions of dollars to get this thing off the ground and do measurements. And if I want to reproduce what John Q. Scientist has done in California, I need to reproduce his laboratory first, which means I need to invest another $10-20 million to get that lab off the ground in order to try to reproduce exactly his measurements. Of course, that's not going to work. So what we tend to do is to look for as many similarities as we can in an experimental setup, and test those to see how they behave. And that's sometimes easily done and sometimes not. And of course, in different fields, there's going to be a different price tag on these things. So, not everybody's going to build a CERN in order to test, right, the particle physics behavior at high fields.
So if we look at the big benefits here for humanity, if you want to call it, I'm guessing your work would then help affect policies for reducing these not fluorocarbons, but the other fluorinated ones you mentioned. And what are the other big takeaways? Like, are we going to get better medicines, better something? Fill in the blanks there. What's the big goal, the big win?
Yep. So on the personal medicine side, there's wins in the sense of diagnosis and early detection for sure. There's a belief that the body doesn't go off the rails immediately, right? We have internal correction mechanisms and healing mechanisms that try to fix any illnesses we might have. And then there's sort of a slow decay into a disease state. Slow on the timescale of molecules, I should say, right? I mean, diseases can go very quickly on the timescale of human beings. So if we can monitor the human chemical space, right, then we have a potential way of diagnosing and catching things early, so that we have a better chance of treating.
And does that have to be through blood tests, or is it going to be saliva, or how do we get the sample fluid or the sample?
All of those things are, all of these things are possible. Blood is a very good way, but it's invasive, right? You need to get the blood out. So things like urine, saliva, tear fluid is a good one, right? You need to sing someone a sad song and collect a tear, and maybe that'll help. So those are directions that we can go in, and ultimately, it would be good for us to map this from an individual perspective, right? Not just as a population perspective, because the way that you metabolize your Friday evening pizza might be different from the way I metabolize my Friday evening pizza. And just because you're showing slightly different concentrations of a particular molecule in your tear fluid doesn't mean that you're ill, right? So we need to take a more detailed perspective of those things. The same goes with developing new pharmaceuticals, right? So new medicines may affect different people differently, and having a more detailed molecular perspective of why that is could be very beneficial, right? So things along those lines are certainly high priorities when it comes to AI in the chemistry space.
Excellent. Well, this is exciting future ahead, I think. All your work is stepwise growing to bigger and bigger impact. This is incredible.
Yeah, we're certainly trying. And the nice thing, of course, is when you're developing machine learning, it's a data handling process that's really, statistics on adrenaline, right? So what we're developing in the chemical space does actually translate to other spaces, right? So recently, we've started working with some optometrists who are monitoring eye movement, and we found that some of the machine learning methods that we've developed for predicting chemical properties actually translate really well into measuring eye movements and predicting neurodegenerative issues, so things like Parkinson's disease. So it's still early days in that realm, but it's all tied together by machine learning and artificial intelligence these days, which is really exciting. That's not something I would have predicted I would have got into in 1997 as that pimply faced little boy who started studying chemistry, right? So it's been fun, and it's really exciting to see where it's going to go.
Okay, well, thanks again. Thank you for joining us, and wish you all the best and continued success in your area of research.
Thank you very much. It's been a pleasure, Harold.
Awesome. Take care.